Hi, good morning. I'm Liangfeng Han. I'm the second speaker of this session. I'm a reviewer in the team of nasal and orally inhaled products at the Division of Therapeutic Performance, Office of Generic Drugs. Today I will discuss the potential of pharmacokinetic bioequivalence studies in detecting regional deposition with orally inhaled drug products, and I will present some data from FDA-funded research projects related to this discussion. This presentation has three learning objectives. First, I will describe the function of the comparative clinical endpoint bioequivalence study in establishing equivalence in local drug delivery. Second, I will discuss OGD's current thinking on the challenges with using PKBE studies as part of an alternative approach for assessing equivalence in regional deposition. Third, I will explore whether PK studies can detect difference with orally inhaled drug products in the lung regional deposition, that is, the central to peripheral drug deposition ratio. For locally acting OIDPS, such as dry powder inhaler and the metered dose inhaler, OGD's current PSG recommendations include the aggregate weight of evidence approach for establishing BE. The aggregate weight of evidence approach includes in vitro studies and the in vivo pharmacokinetic and the comparative clinical pharmacodynamic BE studies, in addition to formulation sameness and the device similarity. Note that a comparative PDBE study with clinical endpoints is the only tool addressing local action in the lungs because the relevance of in vitro and in vivo PK studies to drug concentrations at the local site of action and to BE establishment is not clear. However, the comparative clinical PDB studies are challenging for several reasons, including their lower sensitivity to performance difference because of the flat dose-response relationships for inhaled corticosteroid-containing products, their cost, and the needs for larger sample size and the long study duration, which could be thousands of patients and the multiple week study duration for inhaled corticosteroid products. Although a PK study is generally more sensitive to performance difference, it's currently limited to assessment of systemic exposure since they measure plasma concentrations downstream from the local site of action in the lungs. With that said, there is the potential that PK studies may detect difference in the pulmonary available dose and the pulmonary mean residence time. The potential for PK studies in detecting difference in local drug delivery for use as a tool for assessing local equivalence in the lung has been previously discussed in a 2009 workshop. Recently, FDA posted a draft guidance on beclomethasone dipropionate inhalation aerosol method that proposes an alternative approach to the comparative clinical endpoint BE study, including additional supportive in vitro, in silico, and in vivo PK studies. After oral inhalation, the drug gets into systemic circulation, mainly through the lung, or to lesser extent, through the gut. To further evaluate the sensitivity of a PK study in detecting regional drug deposition in the lung, FDA funded two research projects conducted by a research team at the University of Florida, in which different DPI formulations were manufactured. The objectives of these research studies were to evaluate if PK is sensitive to DPI formulations that differ in central versus peripheral lung deposition ratio, and to perform an in vivo PK study in healthy adult subjects after a single dose of different orally inhaled formulations. In these research projects, the study drug was fluticasone propionate, that is a slowly dissolving API with very little absorption through the gut. The research team hypothesized that a formulation with small particle size will be more likely to reach the lung periphery as shown on the left panel of the figure. In the periphery, there is less mucociliary clearance, and along with its larger surface area, can lead to a faster, more extensive absorption in this region. This may subsequently result in a higher Cmax and AUC. On the other hand, a formulation with a larger particle size will be more likely to reach the central part of lung, as shown on the right panel of the figure. For a larger particle size that lands in the central region of the lung, there is more mucociliary clearance, and with the lower surface area compared to the peripheral region, that can lead to slower and less extensive absorption, which may present as a lower Cmax and AUC. Therefore, the research team hypothesized that PK parameters such as Cmax and AUC may differentiate the central versus peripheral lung deposition of an inhaled drug. The first step of the study design was to prepare 3 DPI formulations with the same amount and particle size for the API, but mixed with different lactose fines. So the resulting formulations would have the same amount of API and lactose, but have different medium mass aerodynamic diameters due to the difference in lactose fines content. Ideally, the formulation would have the same delivered dose and the dissolution rate since the API was from the same batch. As the result, 3 different formulations may differ in lung regional deposition. Next, aerodynamic particle size distribution. Realistic APSD using anatomical throws coupled with inhalation profiles and the dissolution test were performed to characterize the formulations and help control for potential difference in total delivered dose to the lung. Then the PK study and the PK data analysis using non-compartmental analysis were conducted. Here we are showing the 3 formulations that were manufactured. They have the same amount of API and lactose, but the content and the size of the lactose fines were different. As a result, formulation A has the largest medium mass aerodynamic diameters and potentially more likely deposit in the central region. And formulation B and C have smaller and similar MMAD and potentially more likely deposit at the lung periphery. APSD test was conducted using next generation impactor at a flow rate of 60 liters per minute. The data showed similar drug deposition at stage 2 and 3 across the 3 formulations, but a smaller amount of drug deposited on stage 4 to 7 and the microorifice collector for formulation A. This data is expected as formulation A had the largest MMAD. Next, the in vitro dissolution rate was measured using 2 methods. The first method used was the transwell insert method, conducted by the University of Florida research team. Formulation A has the longest mean dissolution time, about 15.4 hours, while the formulation C has the shortest time, about 10.3 hours. So formulation A has the slowest dissolution rate. The second method used was USP apparatus 5 pedal over disc method, which was conducted by our collaborator from University of Bath. We got the similar result. Formulation A has a slower dissolution rate compared to formulation B and C. Another in vitro characterization method used was the more realistic APSD study, using a combination of 3 mouth-throat models and 1 inhalation profile. The 3 models are VCU, AIT, and OPC. The goal of these tests was to determine the total lung dose for each formulation. When results were obtained with the 3 models and compared directly, the absolute amounts and the ratios between the DPI formulations differed between the different models. To account for potential difference in the lung doses received by the healthy volunteers in the PK study, the research team utilized an average ratio for dose normalization during the PK data analysis. The ratio is 1 to 1.3 to 1.2 for formulation A, B, and C, respectively. After the in vitro characterization was completed, the PK study was conducted. The PK study design was a single-center, double-blinded, randomized, 4-week crossover PK study in 24 healthy adult volunteers. Subjects inhaled one dose of 500 microgram fluticasone, that is 5 capsules of 100 microgram fluticasone, using the modified Plastape inhaler. Individual inhalation profiles were recorded to ensure the inspiration peak pressure and the flow rate of each subject was within normal range. The assay sensitivity was 1 picogram per ml, and the PK data were analyzed using non-compartmental analysis. Here we see an illustration of the PK study design. The 4 treatments were formulation A, B, C, and CR, which was the replicate of formulation C to assess intra-subject variability. The washout period between treatments was 5 days, and the blood samples were taken within 30 minutes prior to the dosing and at as early as 5 minutes and up to 24 hours after last inhalation of the 5th capsule. Before dose normalization for potential difference in the lung dose, the mean plasma concentration were near identical for DPI formulations B, C, and C repeat, which led to closely comparable PK parameters for these treatments. But the formulation A achieved considerably lower concentrations during the first 3 hours, and it had a smaller Cmax and AUC. As formulation A has the largest MMAD and was potentially more likely to deposit in the central lung, the lower Cmax and AUC was consistent with the research team's expectations. After dose normalization, formulation A still had a smaller Cmax compared to formulation B and C. However, its AUC was similar with the other 2 formulations. This suggested that the Cmax parameter may be a PK parameter that is sensitive to lung regional deposition. Of note, difference in the dissolution rate in different formulations could only explain a small portion of this larger observed difference in the mean Cmax. Therefore, difference in dissolution rate did not change the research team's conclusion that Cmax may be a PK parameter sensitive to lung regional deposition. As I mentioned before, this PK study suggested that Cmax may detect different regional deposition between different formulations before and after dose normalization, while AUC appeared to detect different regional deposition only before dose normalization. So this PK study suggests that Cmax difference rather than AUC difference may indicate difference in regional deposition. In conclusion, the conducted PK study demonstrated that the PK can detect the formulation difference, lung dose difference, and the pulmonary residence time difference. This study suggests that the Cmax difference may indicate a difference in lung regional deposition. But after dose normalization, AUC failed to detect the difference in lung regional deposition. Therefore, this research study did not fully support the conclusion that the PK can identify difference in lung regional deposition ratio when analyzed by non-compartmental analysis methods. We also find that the difference in lactose fines that impacted MMADs were able to alter in vitro performance and the in vitro dissolution profiles. This difference in product performance were detectable with Cmax and AUC, although the relationship with these metrics and the regional deposition still requires further study. Before ending this presentation, I would also like to leave you with some of the lessons we learned through our collaboration with the research team. First, selection of the mouse throat model may be critical for estimating the in vitro total lung dose, and so included as part of the considerations for how to control for potential difference in delivered dose between formulations, such as with dose normalization. Efforts should be made to reduce potential variability when conducting the PK study, such as proper staff training, study design, number of doses. The results from this study showed that PK parameters may be sensitive to differentiated regional drug deposition. This may be product dependent, and the sensitivity may vary between different PK parameters. Importantly, this research study illustrates just one example for how a PK study may be designed to evaluate its sensitivity in detecting regional drug deposition between different products. If you have a different study design that you believe is scientifically justified, and you wish to include it as part of your alternative B approach to conducting a clinical endpoint study, the agency highly encourages you to submit a pre-end product development meeting request. Finally, I would like to thank Dr. Hauschhaus and Dr. Brita and their research team at the University of Florida, along with their collaborators at the University of Bath and the Virginia Commonwealth University for doing excellent work in conducting this research. I also thank my colleagues from Office of Research and Standards and the Office of Testing and Research shown here for their comments and suggestions from initiation to completion of the research project. Finally, I have one challenge question for you. The CCEPB study is included in the weight of evidence approach because it may provide information regarding A. Safety, B. Efficacy, C. Equivalence in local drug delivery. After 5 seconds, I will reveal the answer. The correct answer is C. Thank you for your time. That wraps up my talk. I will turn to the next speaker. I will come back later in the QA session for more discussion.